Okay, I welcome you all today in today's class of 601 Advanced Molecular Biology. So today we'll be studying miRNA and SiRNA. So these miRNAs are non-coding RNAs. Okay, and they usually are 21 to 24 base pairs long, and they are found in plants, animals, and viruses. So their main function is RNA silencing and post-transcriptional regulation of gene expression. So they regulate expression of various genes. Okay, in plants usually they are needed for development, metabolism, and stress response. These miRNAs usually code against transcription factors because if there's a pathway, say gene 1, gene 2, gene 3, gene 4, for example, say characterized by SNP pathway or cold responsive pathway. So they are controlled by, if this is a cold responsive pathway, they will be controlled by CBF, C repeat binding factor. How can I change this to mirror image? Because I think the video is coming, the board is coming a mirror image on the screen. Do you see mirror image?
तो क्लियर नहीं दिख रहा है.
ये क्या लिखा मैंने? ये
बी
सीधा दिख रहा है कि उल्टा दिख रहा है?
सर अब ठीक दिख रहा है, ऐसे ठीक दिख रहा है. सो
Okay, so if we control this transcription factor, we'll control the whole pathway. So that's why these miRNAs mostly control the pathways or the transcription factors. These miRNA genes do contain introns, and they are translated by RNA polymerase 2. Okay, how many polymerases are there? RNA polymerases?
3.
3. Good. So RNA polymerase 2 transcribes these miRNAs, and they are capped, polyadenylated, okay, and spliced. स्प्लाइसिंग भी में होती है, पॉलीएडिनेशन भी होता है और कैपिंग भी होती है. कैपिंग किस एंड पे होती है?
5
और पॉलीएडिनेशन
3
गुड. So if you look at the mechanism of miRNA action, so you have this DNA double stranded. It gets transcribed by RNA pol 2, and you have your miRNA synthesized. This is the 5' cap, this is the poly A tail. Okay. Then what happens? These miRNAs have complementary sequence due to which it forms a hairpin type loop. Okay. Now you should understand all this is happening in the nucleus of the cell. So your RNA polymerase is true that transcribes these miRNAs, and they, due to self-complementarity, they form a hairpin loop, and this loop-like structure, hairpin-like structure, is recognized by DGCR8. This is the protein DGCR8. It recognizes this loop. And it binds to this loop-like structure. And then this whole structure, the DGCR8 and this miRNA, is identified by another protein called as DROSHA. This is the main enzyme that cleaves this hairpin loop here. Okay. And the whole complex is called as a microprocessor complex.
So repeat.
You have this RNA polymerase 2. It transcribes these miRNAs. Okay. It has a 5' cap and a 3' tail and introns that are removed by splicing. Then this miRNA is the loop is self complementary. Means if, for example, you have two strands of DNA, they are complementary to each other. You have a here, you have a T here, you have a G here, you have a C here. These two strands are connected with the help of hydrogen bonds. G and C has 3 hydrogen bonds, A and T has 2 hydrogen bonds. But RNA is usually single-stranded. But they have these sequences that are complementary to each other. So what happens if, for example, this is an RNA strand, okay, this sequence is complementary to this sequence, so they will self-anneal. We call this self-complementarity. Okay, so they form a double-stranded RNA structure. This RNA structure is identified by your DGCR8 protein. This protein comes and binds with this complex here, and this DGCR hairpin complex is identified by another enzyme, DROSHA. Okay, and this DROSHA cleaves this complex here, and you have this, and so this is cleared here. Clear? Now this is called as precursor microRNA. And what happens after this? There's a protein called as exportin. This whole thing was happening in the nucleus. So this exportin protein transfers this pre-microRNA out into the cytoplasm. So now we are talking about the cytoplasm. So this is your pre-microRNA, and then it gets associated with exportin 5. Exportin 5 transfers or translocates this miRNA into the cytoplasm. Now this is your cytoplasm. Now if you see closely, the structure of this will be like what we call as precursor microRNA. Now there is a protein called as dicer that identifies this stem loop. This is the stem, this is the loop structure. Identifies this stem loop. Okay. So once the dicer interacts with the precursor microRNA, it cleaves this loop out. And you have this structure left. After this, what happens? There's a gene called as, or a protein called as AGO2. This AGO2 recognizes dicer and this RNA structure, pre-microRNA structure. So the function of this dicer is to cleave the loop, and then it translocates this RNA to the AGO. Now you have this RNA structure and the AGO structure. This AGO2 makes this RNA single-stranded. Okay. So this strand is released and is degraded, and you are left with one strand of microRNA, and this microRNA strand is complementary to the gene that it has to silence. Okay, so now you have AGA2 associated with your one strand of microRNA, and this RNA strand we call as a guide RNA. Now what happens is this AGO now complex AGO guide RNA complex interacts with RISC RNA induced silencing complex. RISC RNA induced silencing complex. This is the main complex to which your RNA is single-stranded RNA or guide RNA is attached. And there is now the bifurcation of this pathway. What happens? This can lead into two things. Number one, what will happen? Can I rub this? Now you have a risk guide RNA and risk complex. This guide RNA and risk complex binds to your messenger RNA that needs to be silenced. Okay, it binds. And how it binds? The complementary sequence, the guide RNA sequence is complementary to this a sequence in your mRNA. So this is mRNA, this is your guide RNA risk complex. Now the sequence of this guide RNA is complementary to a sequence in the messenger RNA. The messenger RNA that needs to be silenced. Now this risk factor degrades this RNA into two. Okay, and so this is again destroyed by various RNAs in the cell, and the gene is silenced, or the mRNA is silenced. Other pathway is that what we call as occupancy. Since this risk factor and guide RNA is occupying a place on the messenger RNA, and for translation of this messenger RNA into the proteins, you need the ribosomal complex. Now this ribosomal complex cannot be at the same position in which you have this guide RNA risk complex, because it is already occupying the position in the messenger RNA, what we call as occupancy. So it does not allow the ribosomal ribosomes to bind to this RNA, and so there is no translation of the gene. So no translation of mRNA into proteins. So again the gene is silenced. Okay, so do you understand the process? No?
Yes, sir.
So next we have the siRNA. Now there is a similarity in the process of this miRNA and siRNA. So the plants also have siRNA. So the origin of these siRNAs can be transgenes. If we want to silence a gene, we'll incorporate or transfer this siRNA. Into the cell, and we can knock down our gene of interest. Or we can put this siRNA in a transposon, and this transposon we can express in a cell, and once this gets expressed, it will silence our gene of interest. So the main difference between siRNA and mRNA is that miRNA targets the genes other than itself. It does not target itself. However, siRNAs target the genes that produce them. Okay, this is the miRNA difference between miRNA and siRNA. Rest is same. They are 20 to 24 nucleotides. Okay, both interact in the same fashion, but the main difference is that microRNAs target other genes than itself, while as siRNAs target the genes that produce them. So, but there are genes, siRNAs, that inhibit the transcription or inhibit undergo gene silencing or bring about gene silencing. They are called as trans-acting siRNAs. These are the siRNAs that can act on the genes other than the genes that produce them. Okay.